Please welcome our next speaker. So, Dan Gibbs from Cirsium.
I'm Dan Gibbs. I'm the founder and CEO of Cirsium Biosciences. So really what I want to talk to you today is how we can bridge the gap between the breakdown where genetic agency fails, moving to Cirsium's solution in terms of addressing a bottleneck in biomanufacturing in particular, and advanced genetic agency through innovation in manufacturing of AAVs in particular. I want to start with a very quick story that a lot of people in this are familiar with, which is a patient family where we have LGMD2D that was really on track for commercialization. You have families and patients who have been waiting for this treatment, and suddenly that opportunity was taken away from them, really not because the science was lacking, but because the macroeconomic environment and business decisions led to a cancellation of a program. And a large part of that was actually around CMC issues and around cost of manufacturing as well as other clinical progress. So this is really where the rubber hits the road. And this is where our company looked to say, how can we address this, at least focusing on one bottleneck? Because connecting gene delivery to the patient is we have all of these axes, or you've heard a lot about this morning. And really, we're focusing on the manufacturers and the technologists and developing a platform manufacturing technology solution to dramatically lower the cost, speed, and scalability of manufacturing of AAVs and ultimately other complex biologics. So as we look at these bottlenecks, you have this attrition of therapies as you go through the development cycle, ultimately really depriving the patients. And we're looking at, you've got production variability, development timelines, scalability risks, and ultimately the affordability of these therapies. So we position our solution really at the point of manufacturing to dramatically lower the cost. So when we've taken a very innovative approach to do this, and rather than looking at conventional manufacturing, we've looked at alternative manufacturing platforms. And we manufacture AAV in high-density agriculture. We use molecular farming in tobacco plants. And there's a number of advantages to this process. So we have a seed train of plants that are effectively propagated, germinated, grown to a certain developmental state, and they're now ready to be converted transiently into producer plants. And we do this in very high-density vertical farms in a very automated and controlled way. If you're ever in San Diego, please come and visit our pilot facility that is going online by the end of the year. In parallel, we're growing up bacteria called agrobacteria, which is where we have transformed them with the packaging construct. So we grow up multiple strains of bacteria, very, very cheap fermentation, very, very cheap cost of goods on all of this, and a very significant reduction in the overall OpEx and CapEx of the process. At a particular developmental state, we combine these. We take vacuum infiltration and we infiltrate the bacteria into the leaves of the plants. This is a transient, flexible system. Ultimately, we are also going to look at transgenic plants as stable producer plants, but that's certainly a way away. And then about 5 days after we infiltrate the plants, we can harvest them. We can take them through standard, scalable clarification and ultimately into fill finish. So harvest and filtration is, again, fully automated. We make AAV smoothies. And then we can take that through purification processes that everybody here is very, very familiar with. Once we get to a cleared lysate, our product is compatible with really all of the standard forms of purification of AAV. This then goes through quality control and into fill finish. So this really has a measurable impact in these 5 key areas around production timelines. We have very high therapeutic quality. The cost of goods is dramatically lower, and patient access ultimately will benefit from these significantly reduced manufacturing costs, as well as the speed of process development is dramatically shortcutted because this is a scale-out approach. So in using this pressure point of manufacturing, we really hope to address all of these axes to provide a much cheaper, scalable, and globally available therapeutics to the patients. That's our ultimate goal. We're working with a number of early adopters to develop this process through to ultimately IND-enabling studies towards the end of 2026 into the beginning of 2027. So obviously, it's a brand new platform. We get a lot of questions about, well, okay, what does their AAV that comes from a plant look like? We have very, very good comparability data. So we've produced material in HEK cells and compared it to plant-produced material. This is AV9 carrying a GFP. We've also produced some BBB crossing capsids and recently heard from a collaborator at the Salk that they've got marmoset data with that, and it's looking very promising. We have very comparable quality metrics. This is for a finished drug product. And just looking at your sort of stunner data that a lot of people hear from plant-produced AAV compared to HEK cell-produced AAV, looking at the particle density and the capsid intensity. So we've taken this in vivo. This is just the study design for a small, both IV and intrathecal administration into African green monkeys with a 6-week in-life period and a vehicle control. These are all immunologically naive at the time of dosing. Very quickly, we see increased transgene expression across a number of target tissues. So the green bars are the plant-produced monkeys dosed with a plant-produced AAV are normalized to the HEK cell-treated group, which is the red line. Skeletal muscle especially, as we see increased transgene expression from very similar biodistribution. And that could be explained by some of the additional data that we see is throughout the in-life and at the end period, we see a significant reduction in neutralizing antibody responses to the plant-produced AAV compared to the HEK cell AAV. And this, we're interested in the mechanism of this, of course, but it does not seem to be driven by changes in PTMs, which is another question that we get asked a lot, is, how does you're producing this in a non-mammalian system, how is the capsid being modified? So we screened an entire range of post-translational modifications, not just glycosylation. We really looked at everything and saw no significant differences. I'm just highlighting glycosylation here. Again, on AV9 as being one of the major PTMs that people look for. So overall, I want to bring it back to the patients that are really not having genetic agency right now and how a significant step change in manufacturing in terms of speed, cost, and scalability can dramatically improve their access. And that's really where a lot of our early adopters are looking at taking a risk with us going into IND, particularly in the rare and ultra-rare disease space. So I'd encourage anybody that is interested in learning more about our platform, please come talk to me. And Adam is our head of BD here, and we're happy to discuss this in a lot more detail. Thank you very much.
Thank you, Dan.